something survived a nuclear blast. Not a bunker, not lead shielding, not a machine built to withstand it. A bacterium. Tiny. Pink. The size of nothing. Found living inside a tin of irradiated meat in 1956, after scientists fired enough radiation at it to kill a human being 1,000 times over. It was completely unbothered. The Guinness Book of World Records lists it as the most radiation-resistant organism ever discovered on Earth. Scientists spent 70 years trying to understand how, and the answer, when it finally came, did not just explain one strange bacterium. It opened a door to changing how we treat cancer, how we protect astronauts in deep space, how we think about the limits of life itself. I am going to show you exactly what is happening inside this cell, in a way you will not forget. Before we get to the science, 30 seconds on what this thing actually is. Deinococcus radiodurans is a bacterium. Microscopic, pink. It lives in soil, in dust, in dried food, in hospital instruments. It is probably on your skin right now. It does not make you sick. It has no interest in you. It is completely harmless. It just exists, quietly, everywhere, carrying inside it one of the most extraordinary survival systems ever evolved on this planet. Now here is the first strange thing. When most bacteria divide, they split into two. Simple, clean. This one splits into 4, simultaneously. And those 4 cells stay attached, forming a perfect little square. Its pink color is not random. It comes from a pigment that absorbs ultraviolet radiation before it can reach the cell interior. The color itself is a shield. And this organism carries between 4 and 8 complete copies of its own genome. Most bacteria carry 1. This one keeps 8 backups, like a system so important it cannot afford to lose the data. One more thing. A team of scientists once proposed, in a published paper, that this organism may have evolved on Mars and arrived here inside a meteorite. Most scientists disagree, but the fact that someone looked at this organism and thought, this could be extraterrestrial, tells you everything you need to know about how strange it really is. For decades after its discovery, scientists had a working theory. This bacterium must be protecting its DNA somehow. Some kind of biological armor around the genome. A shield. Something that prevented radiation from breaking the genetic material in the first place. Logical. Reasonable. Completely wrong. Here is what radiation actually does to a living cell. Ionizing radiation, the kind used in nuclear reactors, in cancer treatment, in the experiment that found this organism, does not gently damage DNA. It snaps it, clean through. At 5,000 Gray, the dose this bacterium routinely survives, its DNA shatters into over 200 fragments. Every chromosome, every gene, broken into pieces. In you, 5 Gray is lethal. That is it. 5. This organism survives 1,000 times that. So scientists searched for the shield. They looked for special proteins protecting the genome. They mapped everything. They found nothing. Because the DNA was not being protected. It was shattering. just like in every other organism. The shield was somewhere else entirely, and nobody had thought to look there. In 2009, a scientist named Michael Daly published a paper in Nature Reviews Microbiology that changed the direction of an entire field. The finding was 3 words: not the DNA. It is not the DNA that is protected. It is the proteins. Here is what that means. When radiation hits a cell, 2 things happen simultaneously. The DNA breaks, and the radiation creates reactive oxygen species, tiny, unstable molecules that tear through everything else. The enzymes, the repair machinery, every tool the cell needs to survive. In most organisms, both happen at once. DNA shatters, and the proteins that would fix it are destroyed at the same time. No tools, no recovery. Death. In Deinococcus radiodurans, the proteins survive intact. Picture this. A vast library burns completely to the ground. Every book destroyed, every record gone. But all the librarians walked out, unharmed. And they remember everything. Every book, every shelf, every page. They rebuild the entire library from memory in 6 hours. That is what happens inside this cell after a radiation hit. The repair proteins, perfectly intact, reassemble 200 DNA fragments back into a working genome. But how are the proteins protected? In December 2024, a team at Northwestern University published a paper in one of the most prestigious scientific journals in the world. They found 3 molecules working together: manganese ions, phosphate, small peptides. Individually, each offers modest protection. Together, they form an antioxidant shield that absorbs the oxidative damage before it can reach and destroy the repair proteins. The combined power exceeded everything the researchers had predicted. Lead researcher Brian Hoffman described his reaction to the data in 3 words. Oh my God. The 3 molecules wrap around the cell's repair machinery and take the hit, so the repair proteins do not have to. The librarians are protected, so they can save the library. So why does a bacterium in a tin of meat matter to you? 3 reasons. The first is cancer. Radiation therapy is one of the most powerful tools we have against cancer. It works by destroying the DNA of tumor cells, so they cannot divide and spread. But healthy tissue surrounding the tumor gets hit too. Patients suffer serious damage to organs and healthy cells that had nothing to do with the cancer. It is one of the biggest limitations of modern treatment. Now imagine this. What if we could give healthy cells a version of this protection before treatment, so they survive the dose that destroys the tumor? Cancer cells, unprotected, destroyed. Healthy cells, shielded, walk out intact. Research is active, not in clinics yet, but the mechanism is now understood. The direction is clear. The second reason is space. Anyone traveling to Mars faces months of cosmic radiation exposure, radiation that Earth's magnetic field normally blocks. On Mars, there is no such protection. The 3-molecule complex is being studied as a potential radioprotectant for human cells on deep space missions, something you take before exposure, not after. The third reason is nuclear waste. This organism has already been genetically engineered to survive inside nuclear waste sites, environments lethal to everything else, while simultaneously breaking down toxic compounds. A living machine, deployed where nothing else can go. 3 problems, 3 fields. 1 pink bacterium from a tin of meat in 1956. Here is what we know. A bacterium discovered by accident in a tin of irradiated meat carries a survival system so precise and so elegant that it took humanity 70 years to understand it. Its DNA shatters every time into hundreds of pieces, but its proteins survive, and its proteins rebuild everything. The key is 3 molecules that nobody predicted would work together. And now, for the first time, we understand the mechanism well enough to ask the question that actually matters. Can we give this protection to human cells? Not the organism, the shield. If the answer is yes, cancer treatment changes, space medicine changes, how we think about radiation entirely changes. Researchers are working on this right now, in multiple countries, in multiple labs. Every source used in this video is linked in the description. Nature Reviews Microbiology, PNAS, Cold Spring Harbor Perspectives in Biology. Every claim is traceable. Every paper is accessible. One question before you go. If this protection could be packaged as a treatment, something you take before cancer radiotherapy, to shield your healthy tissue while the radiation destroys the tumor, would you take it? Or would you worry that protecting cells that should not be protected might cause problems we cannot yet predict? There is no right answer, but the fact that we are even asking this question because of a spoiled tin of meat in 1956, that is what science does. Case 0. Every mystery begins somewhere.